Right now, running through your veins, is a molecular signature that has decided, over the last 10,000 years, who lived through a cholera outbreak and who didn't. Who caught malaria and shrugged it off, and who didn't survive the fever. It's for letters. O, A, B, AB. You've probably had it printed on a donor card or a dog tag at some point in your life, and you've probably never thought about it again. But that tiny classification sitting on the surface of your red blood cells has been quietly shaping human survival for millennia, and in some cases, it's still deciding outcomes in hospital rooms today. So here's the real question this raises. If blood type is just a footnote on a medical form, why do scientists keep finding it tangled up in malaria resistance, heart attack risk, mosquito bites, stomach cancer, and even the outcome of the Black Death? Why does a molecule that seems to do nothing more than tag your blood cells keep showing up as a survival variable across completely unrelated diseases, in completely unrelated corners of the world? That's what we're going to dig into. And fair warning, it gets weirder the deeper you go. Here's what most people take for granted. You know your blood type, or you think you do, mostly because you'd need it in an emergency. O is their universal donor, AB is their universal recipient, and Rh positive or negative determines whether you get a plus or minus sign after the letter. That's the entire mental model most of us carry around. For letters, 2 signs, done. What almost nobody carries around is the fact that this system exists because early 20th century doctors kept watching blood transfusions kill people for reasons nobody could explain, and what they eventually found underneath that mystery turned out to be a map of humanity's oldest war, the one against infectious disease. Start in Vienna, in the year 1900. A pathologist named Karl Landsteiner is working in a lab at the University of Vienna, mixing blood samples from his colleagues in test tubes, just to see what happens. This wasn't glamorous work. He was pipetting serum from his own co-workers, some of it his own blood, and watching under a microscope as certain combinations clumped into gritty, ruined clusters while others stayed smooth and mixed. By 1901, he had sorted people into 3 groups, A, B, and C, which would later be renamed O. A 4th group, AB, was identified by his students 2 years later. Landsteiner had just explained why transfusions, which doctors had been cautiously attempting since the 1600s, sometimes saved a patient and sometimes killed them within minutes. He won the Nobel Prize in 1930 for it. Here's the thing though, Landsteiner thought he'd solved a plumbing problem. He had no idea he'd just uncovered a battle scar from thousands of years of infectious disease pressing on the human genome. Move to West Africa, and the mechanism starts to show its teeth. There's a blood group system separate from ABO called the Duffy antigen, and across huge stretches of sub-Saharan Africa, somewhere around 90 to 97% of people carry a mutation that makes their red blood cells Duffy negative. That mutation does one very specific thing: it removes the docking point that Plasmodium vivax, one of the parasites that causes malaria, needs to break into a red blood cell. No docking point, no infection. Populations in malaria-heavy regions were essentially put through a multi-thousand-year filter, generation after generation, where carrying that mutation meant a meaningfully better shot at surviving to have children. Here's where it gets interesting: this isn't ancient history playing out in a museum. Duffy negativity is one of the fastest, most complete instances of natural selection ever documented in humans, and it happened because of a single-celled parasite carried by a mosquito. Now jump to the Ganges Delta in Bengal, 1817, the starting point of the first true cholera pandemic. Cholera tore through Asia, into Europe, and eventually the Americas, killing in a matter of hours through catastrophic dehydration. Researchers looking back at cholera mortality patterns, and later studies on Vibrio cholerae toxin, found something striking: people with blood type O were dying at dramatically higher rates, in some outbreak studies experiencing severe, life-threatening cholera up to 7 times more often than people with other blood types. Their mechanism traces back to how the cholera toxin interacts with the gut, binding more efficiently in people whose cells carry the O antigen structure. Sound familiar? Because this is the same theme as the malaria story, just flipped. In one case, O offers no advantage. In cholera-soaked river deltas, it may have been a liability, quietly pushing survival odds toward A and B carriers in exactly the regions where cholera was most relentless. Here's another angle nobody expects: mosquitoes have preferences, and they are not subtle about it. A widely cited study out of Japan, led by entomologist Yosuke Wedding-associated research teams in the early 2000s, tested attraction rates by exposing mosquitoes to different blood types in controlled outdoor trials. Type O individuals got landed on and probed nearly twice as often as Type A individuals. The leading explanation involves secretor status, a separate genetic trait where about 80% of people actually release their blood type antigens into their sweat and skin. Mosquitoes appear to be picking up on those chemical signals like a hunter reading a scent trail. So if you've ever been at a barbecue getting devoured while the person next to you stays untouched, there's a real chance your blood type just outed you to every mosquito in a 30-foot radius. Then there's your heart, and this one has direct modern medical weight. People with non-O blood types, meaning A, B, or AB, carry roughly 25% higher levels of a clotting protein called von Willebrand factor, along with higher factor VIII levels. Both proteins are central to how your blood clots. A landmark meta-analysis published in the journal Circulation, pooling data across roughly 20 studies and more than 90,000 participants, found that non-O blood types carried around an 11% higher relative risk of coronary heart disease compared to type O. Here's what nobody tells you when they talk about blood type diets and wellness fads: this is one of the few blood type health claims that actually holds up under a mountain of clinical data, and it has nothing to do with diet. It's about clotting chemistry, baked into your genome since before you were born. Stomach cancer tells a similar story from a different angle. Type A individuals show a consistently higher association with gastric cancer, a pattern researchers first noticed in the 1950s and have kept confirming since, most recently tied to how blood type interacts with Helicobacter pylori, the bacterium responsible for most stomach ulcers. H. pylori appears to bind more readily to the gastric lining of people with type A antigens, setting off decades of chronic inflammation that, in a subset of cases, tips toward malignancy. It's not destiny: plenty of type A people never develop stomach cancer, and plenty of non-A people do, but population level, the pattern has held for 70 years of research. Rh factor deserves its own chapter, because this is where blood type moved from statistical curiosity to literally reshaping how pregnancy is managed worldwide. In 1939, a physician named Philip Levine was investigating why a woman who had just delivered a stillborn baby went into a severe transfusion reaction after receiving blood from her own husband, blood that should have matched her ABO type perfectly. Levine, working with Rufus Stetson, traced the reaction to an entirely separate antigen, later named Rh after the rhesus monkeys used in the related research. What they'd found was Rh disease. When an Rh-negative mother carries an Rh-positive baby, her immune system can start manufacturing antibodies against the baby's blood cells, a problem that gets worse with each subsequent pregnancy. Before treatment existed, this killed or severely disabled tens of thousands of infants a year globally. The fix, a shot called Rho-D, immune globulin, wasn't developed until the 1960s. Here's the human detail worth sitting with: Levine wasn't chasing a Nobel Prize when he made this discovery. He was trying to explain one specific woman's brutal night in a delivery room, and it ended up saving a global population of babies he would never meet. And then there's the story that has nothing to do with medicine at all, and everything to do with culture. In 1927, a Japanese psychologist named Takeji Furukawa published a paper linking ABO blood type to personality traits, claiming type A people were meticulous and anxious, type B people were creative and chaotic, type O people were confident leaders, and AB people were complicated hybrids of the other three. There is essentially zero rigorous scientific support for any of it. But in Japan, and to a lesser extent South Korea, the idea never died. Dating profiles list blood type. Job applications have historically asked for it. Companies have organized teams around it. It's a completely unscientific theory that nonetheless demonstrates something very real: humans are wired to grab onto any available marker, even a meaningless one, and build an entire identity system on top of it. Finally, look at how blood type built the modern medical infrastructure you'd rely on in an emergency. During World War II, a surgeon named Charles Drew developed large-scale blood banking and plasma storage techniques, running their Blood for Britain program that shipped thousands of units of plasma across the Atlantic in 1940 and 1941, then organizing the American Red Cross's National Blood Bank system. Drew's work on separating and preserving plasma meant that for the first time, blood type mismatches could be managed at scale, across battlefields, hospitals, and disaster zones. He resigned from the Red Cross in 1942 over its policy of racially segregating donated blood, a policy with no scientific basis whatsoever, since blood type has never correlated with race in any biologically meaningful way. Drew insisted, correctly, that a unit of type O blood is a unit of type O blood, no matter whose arm it came from. So look at what you're holding. You've got Landsteiner in a Vienna lab explaining why transfusions were killing people. You've got Duffy negative populations in West Africa, essentially malaria proofed by a single mutation. You've got cholera in Bengal picking off type O bodies at higher rates in 1817. You've got mosquitoes tracking your blood type through your own sweat. You've got clotting proteins quietly raising or lowering your heart disease risk. You've got H. pylori exploiting type A stomach linings. You've got Philip Levine untangling Rh disease from one woman's delivery room emergency. You've got an entire personality mythology built on top of it in Japan. And you've got Charles Drew turning all of it into an actual system that could save lives at scale, while fighting to strip the racism out of it along the way. Here's the unifying thread underneath every single one of those stories. Blood type isn't a static label sitting quietly on a lab form. It's a live record of pressure, thousands of years of pathogens, parasites, and toxins pressing against human populations in different corners of the world, and the human genome pushing back, unevenly, region by region, disease by disease. Every advantage baked into your blood type came at the cost of a vulnerability somewhere else. Resist cholera, maybe you're more exposed to heart disease. Dodge malaria, maybe a different pathogen finds its opening. There's no perfect blood type. There's only a set of trade-offs your ancestors' immune systems negotiated on your behalf, long before anyone had a microscope to see what was actually happening. Which brings it back to that phrase printed on your donor card, the one you've probably never thought twice about. It's not administrative trivia. It's a message in a bottle from every ancestor you've ever had, written in the language of antigens and antibodies, a running tally of every plague, parasite, and pathogen your bloodline managed to survive long enough to pass itself forward. The next time you glance at those two letters and a plus or minus sign, you're not looking at a medical footnote. You're looking at the surviving half of humanity's oldest, longest argument with disease, still being carried forward, quietly, in you.